the process by which photons, particles of light, are transferred into electrical signals. This happens in the retina of the eye through rods and cones called photoreceptors. Rods are highly sensitive. They can be activated by as few as 6 photons. Photons are the quantum of light. Cones require a significantly larger number of photons to ensure a signal is made. 3 types of cone cells are represented by the response pattern to wavelengths of light. Cone cells reply to light of short, medium, and long wavelengths. This is determined by the absorption of photons. All the wavelengths together create color vision. Opsin is contained in both rods and cones. It is glued to a light-absorbing chromophore. Opsin comes in different types and aids in transducing light of many intensities and wavelengths. For one photoreceptor, there is one type of opsin. Number 1, rhodopsin is found in rods and converts dim light. Number 2, photopsins are found in cones and generate color vision. When light is depleted, photoreceptors are then depolarized to a membrane potential of resting of negative 40 millivolts. The plasma membrane will hyperpolarize the light to negative 70 millivolts. The second messenger molecules accountable for the regulation of a depolarized resting state in photoreceptors is the cGMP, in the nucleotide form of cyclic guanosine monophosphate. Cyclic GMP-gated channels are in charge of the light-induced changes the photoreceptors create in electrical activity. This takes place in the outer segment membrane. Phototransduction involves 3 biochemical occurrences. Opsin is activated when light enters the eyes in the photoreceptors. Rhodopsin brings a decrease in cGMP concentration. Hyperpolarization happens after the exposure to light. Rhodopsin is a G protein receptor that is grouped with opsin and 11 as retinal. Light is stimulated by rhodopsin in the disc receptor generates activation of transduction. GTP-bound alpha subunit of transduction, this stimulates activation of PDE. PDE hydrolyzes the cGMP into only GMP. This decreases the concentration of outer segment and the sodium channels therefore close.